Hello, my name is Chaelynne Lohr, and I'm a second-year master's student with the Department of Forensic Sciences. I'm going to be talking about RNA methylation patterns of forensically relevant transcripts. We have found that RNA degradation can be used to estimate the age of a sample that is found at a crime scene. Further analysis through RNA sequencing shows that degradation occurs faster at the 5' end versus the 3' end of these transcripts that we looked at. This led to the development of the 5' to 3' assay, which quantifies and compares each end of a transcript in a single reaction in order to estimate sample age. This assay has been validated on dried bloodstains. However, why the 5' end degrades faster than the 3' remains unknown. As this phenomenon is being observed in dried samples, we are hypothesizing that chemical hydrolysis reactions are responsible for breaking the RNA molecule, and therefore that chemical modifications of the 5' or 3' ends of the RNA molecule may be affecting the degradation rate. A literature exists that suggests that methylation of RNA molecules can alter the kinetics of RNA degradation through affecting transcript stability and also possibly dependent on the RNA binding proteins that are present. Therefore, we are aiming to interrogate the RNA methylation patterns of 2 forensically relevant transcripts in dried bloodstains. We developed a novel RNA enrichment technique that utilizes DNA probes that are designed to hybridize to either the 5' end or 3' end of a transcript. These enriched products will then be hydrolyzed into nucleotides for analysis via liquid chromatography tandem mass spec. We designed DNA probes that are biotinylated in 120 base pairs in length to enrich the 5' or 3' ends of a transcript, and each end was enriched in separate reactions. We utilize a trizol plus Zymo column kit for RNA extraction. Following that, we perform a hybridization of the biotinylated DNA probes to the RNA for our target selection. We then digest those products with RNase A in order to remove off-target products. And this works because RNase A digests single-stranded RNA, and so it will cleave off anything that is not hybridized to the DNA probe. That product is then digested with DNase 1 in order to remove the DNA probe, and following purification, we are left with the 120 base pairs of RNA that was hybridized to that probe. That 120 base pairs will then be hydrolyzed and analyzed via mass spec. Here you can see some preliminary data for our RNA enrichment of both the 5' and 3' ends. These products were not digested with RNase A so that we could quantify the whole transcript that we were pulling. Bullet data represents where the DNA probe was hybridized to, and the delta CT represents the enriched CT product minus the control CT. Here is some enrichment data following RNase A digestion. We did have some off-target amplification. However, for HBB, you can see that it is very abundant in blood, which is shown by the control. But we are getting a CT of almost 35 in our enriched product, so we're hoping to be able to fix this little bit of amplification with more stringent RNase A protocol. The other off-target amplification we are seeing may be due to our primer designs, which is shown through some negative controls in other experiments, so that is something that we are working through right now. Our ongoing work includes optimizing our RNase protocol to eliminate all off-target amplification and validate this protocol on 2 more probes for a second transcript in blood. We also need to validate our hydrolysis protocol before sending our samples for mass spec. In conclusion, our preliminary results show that this approach can enrich for our selected target over 150,000-fold. We anticipate observing differences in RNA methylation patterns between the 5' and 3' ends of our selected transcripts, potentially explaining the differential degradation rates of the 5' and 3' ends of RNA molecules. And here are my references and my email if you have any questions. Thank you.